Thank you again, Hakim. What the data I showed you in the first session was not randomized, it was observational. So there is selection bias, and you cannot really compare the outcome between the two arms. So the question came to us, can we actually randomize patients directly in the registry? And I'll tell you about the story. So the vision here is to create what has been referred to as the learning healthcare system. The learning healthcare system is a process of informatics, but it includes incentives and the culture. And I think that cultural aspect is very important. We need to make doctors and healthcare personnel motivated by the culture of improving the care, not only diagnosing and treating, but also improving. And so you feed that circle with new information, you get new questions, you feed that back to the patients and get informed how we can improve quality of care. And that should be embedded in the delivery process in everyday care. So this was really obvious to me when I was in a guideline committee looking at the evidence for thrombus aspiration. And at the time, it was so intuitive for me to suck out the clots. If an artery is closed by a clot and there is a device you can suck it out, of course you should suck it out. You should retrieve it. And in fact, as you can see, about exactly 50% of the patients in Sweden were receiving thrombus aspiration. So it was really varying from 0 to 100% between different hospitals. So from the patient perspective, it was still already at random because it was completely dependent on which doctor you happen to see. You would receive or not receive thrombus aspiration. So why not just randomize from the beginning? And so the evidence for thrombus aspiration at the time was based on one trial looking at EKG changes. It was not designed or powered to look at mortality, but it did show a reduction in mortality, a dramatic reduction, 50%, which was larger than anything else we do in cardiology. It was larger than bringing the patient in an ambulance or taking the patients to the cath lab. So many of us wondered, is it possible to reduce mortality by 50% by one intervention? So we did a retrospective observation in the SCARS registry, looked at all patients in the country, 20,000 patients who had received or not received thrombus aspiration, and we found the opposite. 20% higher mortality in those who had received thrombus aspiration. So it was frustrating. Which is the truth? The small randomized trial or the non-randomized observation, but in all patients in the country? So we realized we need to randomize many patients and realize perhaps the registry can help us. With the registry we can identify all patients, we can collect all the information we need, we can collect endpoints, and we can randomize. So we can do everything in the registry. And so we created what we now call a registry-based randomized clinical trial. It's basically a randomized trial, but we use the registry to be able to conduct the trial. And thereby we combine the two different worlds of observational studies and randomized trials. And in the middle, we have this registry-based randomized clinical trial, which can be a very easy, pragmatic trial enrolling all patients, but randomize them, and thereby we can draw causal inference. And so we just programmed the registry, and so the registry helped us to identify patients. All patients who were above the age of 18, who had a myocardial infarction, was about to do a primary PCI, this red line popped up. This patient may fit into the trial. And there were only two boxes to click. Consenting, and the consent form was provided on the right-hand side in front of us in the cath lab. And the second was to check EKG to make sure it was a true ST elevation myocardial infarction in that region of the heart. And that was it. That was the trial. There was no follow-up, no paperwork, nothing. And that was the reason. The simplicity of it was the reason that we could do, at the time, the largest randomized trial with any device in cardiology that had been performed, 7,244 patients in such a small country, which was 80% of all patients in the country who were eligible for the trial. And the results were important. It didn't show any benefit of thrombus aspiration. In fact, a trend towards harm in terms of more stroke. And we published the results in New England Journal of Medicine twice. But even more important, I think, was the discussion about the methodology, how we did the trial to randomize in the registry. And Mike Lauer, who was the chief of the National Institute of Health in the US, wrote this editorial that perhaps randomizing in the registry is the next disruptive technology or technology in clinical research. And at the same time, we were fortunate that another group in Canada was doing a similar, but a site-based traditional trial, and we were performing these at the same time. And as you can see, the difference, we started enrolling at the same time. We used only one small country. They used multiple countries, multiple centers, fairly a complex procedure. We started enrolling at the same time. When they published their first paper, 2015, we had already published 2 papers in New England at the same time. They had only 6 months of follow-up. We had at the time 42 months of follow-up, and we're still following these patients. We can follow them for a lifetime because they're in our registries. And we had the same conclusion, the exact same results, and importantly, the difference in cost was enormous. So I wouldn't say this is a cheap way of doing it, but it's a cost-effective way of doing trials. We can do so many more trials for the amount of money that we have for research and development. So very cost-effective way of doing research. And also, we can check about the adherence to the therapy. We can look at the hospital level, even on an individual physician level, if the physicians adhere to the new evidence. So we could check here before the trial, during the trial, and after the trial, did they use thrombus aspiration or not? And did they adhere to the new results that we learned? And we can see that most of the hospitals did follow the new evidence and stopped using the device on everybody, routinely. We still said that it's okay to do thrombus aspiration in, high-risk selected patients, but on a routine, we should probably not do it. And thereby we closed this loop of evidence generation. We found an idea, we tested the idea in the registry, we designed a trial, performed the trial, synthesized it in meta-analysis with other trials, let it come into guidelines, and then change practice within 2 years. Change practice not only in Sweden but globally. And so the system seemed to work. The next question we asked, is oxygen really needed to treat myocardial infarction? Oxygen in myocardial infarction is caused by no oxygen in the muscle. But to get oxygen in the mouth or the nose does not make the oxygen come to the suffering myocardium because the artery is blocked. In fact, oxygen is a contracting pharmaceutical agent. So we designed this trial, 6,600 patients, to receive oxygen or receive air in the ambulances, in the emergency department, in the cath labs, in the CCUs, and just randomize. And mortality was the primary endpoint. And we showed in a large trial, 6,000 patients, that there was no benefit of oxygen. Immediately, the guidelines globally were changed. We published in New England Journal of Medicine, and it really changed the way we use oxygen. We could get rid of oxygen in ambulances and emergency department. It doesn't help. It's not useful if you have healthy lungs, if you can breathe. The next trial we also tested was, can you go back one slide? The Validate trial. The first time we did if you can help me, technicians at the background, to back one slide. There you go. The Validate trial. No. Here. Okay. So the Validate was the first pharma trial we performed. Now we wanted to understand, is heparin or bivalirudin better for patients with myocardial infarction, STEMI or non-STEMI? So we randomized again in the registry. Here we had a more complex endpoint. We had bleeding, we had myocardial infarction, death, and we showed again in New England Journal of Medicine that bivalirudin was not superior to heparin. So one could question if you really need to use it if it was more expensive. Third trial that I just presented at the ESC, the design, complete enrollment. This is the first trial we've done with a pharmaceutical company designed to be basis for a labeling, an indication with the FDA in the United States. So it's a double-blind, placebo-controlled, sort of complex trial. Again, in the registry in Sweden and the United Kingdom, enrolled 4,000 patients in 2 countries only, 100 sites. the quickest enrolling trial that AstraZeneca has ever sponsored. Very fast enrollment, dapagliflozin versus placebo to see if standardized dapagliflozin post-myocardial infarction would reduce the risk of cardiometabolic endpoints, death, hospitalization for heart failure, diabetes, atrial fibrillation, etc. I hope I will be able to present the results of this trial at the AHA in the US in November. But it's a large endeavor. It was very successfully performed, I can tell. Another interesting for the interventional community was the SweetPad. We randomized patients with peripheral artery disease to receive drug-eluting stents or balloon dilatation. And interestingly here, the FDA wanted us to do an interim analysis to look at the results before we had completed the trials and to see if there was a mortality difference. So they urged us, the authorities urged us to look at the data and publish the results in New England Journal of Medicine, telling that there was no mortality difference. We have just now completed enrollment in this trial, and the trial will collect not only mortality, but amputation, quality of life, etc. So it'll be a very important trial for peripheral artery disease. This is a just recently completed trial. We're testing a new stent technology. I don't know if you're aware of this stent technology. It's called Dynamics. It's a US-based company, produces this. We randomized with the Medtronic Onyx as the comparator to the bioadapter Biodynamics. This stent is interesting because it behaves like a drug-eluting stent. In the first place, when you put it in the artery, it has the strength. But then there are bridges between the metal connectors that dissolve within 6 months. So after 6 months, it dissolves and becomes more like circles. So it makes the artery retain its biological function, the motion. And so the idea here is on a 1 year check that it's non-inferior to, is not worse than the Onyx stent, but in 2 years that it's superior, that it reduces the risk of reintervention. So the first time also, this is an FDA labeling indication approved by the authorities. It's completed enrollment, 2,400 patients just recently. And here's a list of trials that are either ongoing or have been performed in the registry. So you can see the number of patients. So these are 7, 2, 6,000, 4,000, 6,000, etc. You can see the type of intervention and what registry is. It's cardiology, it's general, it's intervention. Most of them are published in very high-ranked journals. It's about heart failure, it's vaccination, it's medical therapy, it's oxygen, it's techniques like IFR, FFR. It's to treat patients with MINOCA, myocardial infarction with non-obstructed coronary arteries, dapagliflozin, it's Takotsubo, it's Helicobacter, post-myocardial infarction. It's about exercise, it's heart failure, it's surgical trials, cardiac thoracic surgery, what types of graft you should use. And onwards, we have a number of stroke trials, we have diabetes, orthopedic surgery, what type of surgery you would need to do, obesity surgery, we have 3 trials ongoing, we have a couple of trials in vascular surgery with external sponsors and internal, we have cancer, and gynecology labor. So a huge number of trials ongoing, and all of them are extremely large. They're more or less pragmatic and easy to do, and I'm sure that these will change and transform the way we treat patients in the future in all these different specialties. So it is possible. We've done this now for 15 years. The strength is that it reduces the workload for the investigators. It's so much easier to do trials for us as investigators. To some degree, it's easier for patients because everything is integrated in the clinical routine. I randomize while I treat the patient, so it's much easier. It minimizes selection bias. It's easier to enroll more patients, hopefully all of them, depending on the design. Is the resource effective? We have a complete screening log because we know all the patients who were not randomized and we can report how they were. We have a web application developed for this that's available for you also to use if needed. We can do cross-national trials, and it's easy to check the implementation of the results afterwards. There are some limitations. You have to have all the variables have to be validated. It's not ideal for rare side effects because those are not well captured in the registries. Rare diseases is not ideal. at least not in a small country like Sweden. And we have to have the same variable definitions in all the sites that are participating. But now we're building EuroHeart. And EuroHeart is an opportunity to come over these limitations. EuroHeart is a network of registries across all of Europe. In that we have a huge unselected population that we can use. Then we can study rare diseases. We have published a set of harmonized variables that EuroHeart sites should adhere to. And we can do AI on that. We are collaborating with the regulating industry so that they can get access to the data. So in conclusion, as a response to the first question, yes, we can randomize in the registries. And we need more trials. We need more affordable, generalizable, informative trials to inform us and our patients. And integrating it into the registries is a way of conducting more large studies. And now EuroHeart, and I would like to spend some time on discussing that with you also. The EuroHeart network is a network of sites, countries that are independent, that save the data in their own country, but we just harmonize the variables, and then we can collaborate on trials. And I'd be happy to do large pragmatic trials with France on devices or strategies or pharmaceutical agents. That would be fantastic. Thank you.
Thank you very much.